B.Saidi, as usual. Let's start without wasting time. We are going to go topic by topic, and then I tell you that expect this, expect this, expect this. Here, let's start with the biodiversity and the classification of microorganisms. When you go to biodiversity and the classification of microorganisms, what you expect here, know how to draw a structure of a virus, structure of a bacteria. We like to ask that, and the structure of a fungi. Use this book we call Distinction Material. It will help you to get all the marks you are going to find in the paper. There is no question which is going to be found in the paper and cannot be answered by this book. We have for grade 10, grade 11, and grade 12. Since now I'm talking about grade 11, yes, at least you need to know, or you need to have it. How do you get it? The description, check in the description, you'll find the link there showing you that get the book. If you have the book, good and well, let me start. All right. Akira, yes, I'm seeing you here. Yes. All right, what do you expect to find in this topic? Structure of a virus. Anyway, we cannot bring the structure of a virus and the structure of a bacteria at the same time. In most cases, we bring one question concerning about that. So, let me show you here. Let me just share this to you, and then sorry, it's not that. Yeah. So when you talk about the structure of a virus, yes, know the properties of a virus, know the properties of a bacteria. We either bring a virus or we bring a bacteria. So you cannot go without knowing. Listen carefully. Properties of a virus, the living properties and non-living properties. Don't forget that a virus is not classified as an organism. No. A virus, it has some living characteristics, and then the non-living ones. Yes, so if it has the living characteristics, for example, the DNA, yes, it is having the DNA, yes, because DNA is a part of a living character. Sometimes you call the genome, yes, it's the same thing. Don't forget that it is either having DNA or RNA, but it does not have both. And then the non-living characteristics, you can talk about it cannot respire, it cannot reproduce without. It cannot reproduce without a host. It can only multiply if it has a host, meaning that it has another cell which is it has another cell where it must multiply from, because it triggers the DNA of the other one and then it multiplies. So please go when you know the structure of a virus. Sometimes we bring it as a macrophage. When talk about a macrophage, we mean this. It is also a virus, but is a virus which attack another virus. So what is a macrophage? Is a virus which attack, not a macrophage, a bacteriophage is a virus which attack another bacteria. Meaning that, so this virus we can bring it in an empty space, like is a virus, normal virus, or we can bring a virus which attack another bacteria, meaning that, yes, we call it a bacteriophage. So if you look at this, still is the same virus as this one, but this one has specific features which make it to be able to drill the bacteria so that it can enter, and then it put its gene into that bacteria, and then it causes that bacteria to multiply. As it multiply, also the virus DNA is being multiplied, and then many viruses are being raised. So what is important here? What is a bacteriophage? Number 2, structure of a virus. Number 3, the properties of a virus. That is the most important thing. Meaning that if you know the characteristics of virus, you have to know the living and non-living. Then when you go to the bacteria, know the structure of a bacteria. Guys, we will not bring two, but don't do that mistake of saying that, okay, me, I'm going to read only one and then you leave another one. No, the one you have not read is the one we're going to bring. Yes, so make sure that you go when you know the structure of a virus and the structure of a bacteria. This is a simple structure of a bacteria. Yes, so I have a feeling that because this is term 1's work, term 1's work, we set it in term 1. Yes, so it means that since we set it in term 1 and we have been trying to test it, even in projects, so I have a feeling that the content of term 1's work is gonna be minimal. You might find term 2's work more than term 1's work, because term 1's work, we have already set it up. We have already set it in different questions. So you might find a lot of term 2's work than term 1's work. Meaning that expect maybe 60% of term 2's work done, and then expect 40% of term 1's work. Hence, you will have fewer questions maybe in this topic of term 1's work. So if we don't bring a virus, we bring this bacteriophage and bacteriophage. If we don't bring a bacteriophage, we bring the structure of the virus. The structure of the bacteria. If you don't bring that, we're gonna bring. Let me show you. We bring a fungi. Yes, this. So among the diagrams you will find, or you might find in this classification, is number 1. This one, don't go without knowing how to label it. This one, the bread mold. Number 2, the structure. This one, yes. Number 3 is this. Those diagrams. So the rest of diagrams, yes, we can bring them, but we might bring it in a different version. But don't forget that I said that this is term 1's work. Term 1's work, so don't expect to have too much of this because we have already set it before. So we might put more effort on term 2's work. Yes. Yeah, let me know if you have a question you are trying to ask, so that I try to explain it better. So these ones are just shapes of the bacteria. Coccus, round, bacillus, if one is bacillus, bacilli if there are many, spirillum if it's one, spirili if there are many, vibrio with cholera, the one which causes cholera, yeah, the one we are hearing about now. Yes, vibrio is also a shape. Vibrio is comma shaped. So we don't usually ask this one too much. So put on effort, but not that too much. Yes. Let's go to the next question. Focus on this. Too much. Yes. This, what I'm saying now, still is gonna be the same story when it comes to your final exam, when they are dividing it into paper 1 and paper 2. Yes. So economic importance of fungi, we also like to ask it. So sometimes when we don't bring economic importance of fungi, we bring the immunity. If we don't bring economic importance, yes, of fungi, we like this than any other economic importance. Yes. So please focus on the economic importance of fungi. If we don't bring economic importance of fungi, we bring immunity. Yes. So you have to know that. Let me show you what I'm saying here. I'm not going to spend too much time, but I'm trying to tell you what you're supposed to focus on much, so that at least you come back when you know at least you have a distinction. Yes. So this is what I'm saying. These are just diseases. guys, when you talk about diseases, if a question concerning about diseases comes, it will focus on, it will bring a passage, and then we ask you. Don't forget that we have different diseases we cover. Yes. So you might focus on malaria, and then another teacher focuses on another disease. But as long as it is being caused by a plasmodium, is being caused by a protista, is being caused by a virus, as long as that disease is caused by that. So these diseases, don't waste too much time. You just have to master. Yeah, those are how, if they bring this question, how do you manipulate it? But chances are few, maybe let me just say 5% of bringing these diseases, because you have a lot of content and then you have fewer marks. So we might not set each and everything. Yes. So ringworms, yes. So if we are bringing about a disease, we bring a disease which is being caused by a bacteria, which is being caused by a fungi, which is being caused by, but it might not be ringworm, because another teacher might be doing another. So it will be a general question, not necessarily meaning that it's gonna be exactly that. So your focus, I think I'm trying to tell you what you're supposed to. I'm going through this book of distinction material. So if you use this table of the diseases, I think it could be a little bit fine. It could be a little bit of fine if you use this table for now, if you have been reading. So you've been reading, but now you have a fewer time for you to focus. So if you use this table, I think it will be fine for you, just in case it has come. Yes, this is the immunity I was talking about. So if we don't bring the economic importance of fungi, we bring immunity. So what is immunity? So the body's ability to try to resist the microorganisms which have entered our body, yes, to fight all the foreign particles which are coming, and they are going to try to disturb our mechanism of defending, is basically, in layman's language, is what we call the immunity. So is the capability of multicellular organism to resist harmful microorganisms, to resist harmful microorganisms. That is immunity. So you have what you call the natural immunity. This is present when someone is being born. And then you have what we call acquired immunity, which develops after exposure to a pathogen. Here comes another question. What is a pathogen? What is a pathogen? You need to know. A pathogen is a disease-causing organism. A pathogen is a disease-causing organism. Is an organism which causes diseases. Yes, if someone is asking me, do you have any sample questions? Yes, I have sample questions. I'm gonna show you. I'm gonna show you the sample questions. I want to first wrap up with this. Then after that, I'm gonna give you the sample questions. So that when I talk about something like that, okay, let me just show you. Structure of a virus. Check how it looks like. If you want this booklet still, the questions and answers, I can still send you, or you can download it from Thundereduc. And then let me show you here. Yeah, so you see how this table I was talking about. But now these are a lot of diseases. This is what I was talking about. But don't expect to have all these diseases, because rabies and AIDS and influenza, all of them are being caused by a virus. We are not going to bring all of them. Remember, one teacher will be focusing on this, another teacher in another school this. So the technique is, we will not bring HIV, or we will not bring, if we bring HIV, it means that HIV, it is going to be a general question whereby you have to read and extract it from the question, so that even if you didn't read it from the book, you are able to answer the question. Hence, less time to be focused, putting on these diseases which are going to be causing too much time of you to be lost. So put less time here, and then focus on those diagrams I've talked about, and then also term twos work more. Why? Because we have already set term one's work. So structure of a bacteria, you see, so know how to label, you see. So these are some. Provide labels A to D. That's why I'm saying that don't go to the paper without knowing the structure of a bacteria. These are some of the things. I'm not saying that it's gonna come, né? But I'm saying that don't go without knowing it, né? Yes. And I'm saying that we will bring one. Either we will bring the one which is virus, the fungi, the bread mold, or we bring a what? A structure of a virus. But this is term one's work, I repeat. Okay, I'll go there when I reach to the flowers. All right. Then the next question is immunity. So I've talked about. Please don't go without knowing the immunity, because the functions of fungi, yes, and then the immunity. Because I said that if you don't bring fungi, we bring immunity. Yes. All right. Let's go. What is a vaccine? So that one also belongs to immunity. Last time we brought a question concerning about in the project. It was about this bacteria, the bacteria, the resistance, and so I don't expect you're gonna find a lot of bacteria. So I expect to find starting from here. Yes. Because I know that here students struggle too much when it comes to biodiversity of plants. So I'm saying that use, if you have, let me divide for you in terms of hours. Let me say that you have 100 hours for you to read. The first topic I've completed, use 10 hours. Ne? I'm just trying to it means that now I have 90 hours for the remaining topics. Then, when you come to biodiversity, these ones are brophytes, pteridophytes, gymnosperm, angiosperms. Let me tell you how you're supposed to explain when it comes to this. In most cases, what we expect to find here, first of all, what we like is the properties of this. These ones are the simplest organisms in terms of plants. These are the next, and then these are next. These ones, they require water for reproduction. Yes. They don't have seeds. They don't have stems. They don't have leaves. So we call them thallus. They are thallus. They are thallus plants. They have leaf-like, stem-like. When we come to this, we have different examples. We like a moss. A moss. I don't know why. I don't know. Why do we like a moss? I've been the people who have been teaching, you know what I'm saying when I come to this structure of a moss. You know exactly how much I emphasize. And sometimes I even frown trying to tell you that you can't go to the paper without knowing a moss. I don't know why do they like a moss. The way I'm saying it now is the same way I've been saying it, and is the same way I will say it. I don't know why do they like a moss. Ne? I don't know. Ne? But make sure that don't make a mistake. Don't make a mistake unless you know what you call a moss. Yes, this moss, guys, go to the paper when you know it. Number 2, what you need to know about this is this. Phylogenetic tree. Don't go to the paper without knowing this. Do you know why do we like this phylogenetic tree? It's because we have this in grade 12. But we have a phylogenetic tree in a different version. We use different organisms. So make sure that you go when you know what you call a phylogenetic. Let me show you some questions which we can ask you. Oh, yeah, some question like this. Yes, some question like this. Oh, like this. So let me see. Provide a definition of a phylogenetic tree. Phylogenetic tree is a diagram that shows evolutionary relationship among organisms, among a group of organisms, or among organisms of the same kind. Then now here we are finding that state whether the following statement is true or false. So here they give you questions, but they're going to ask you to find the common ancestor of this, or which one. Okay, for example, let me show you here, because some people, they don't know how to use this phylogenetic tree. This one is the common ancestor of all, meaning that all these ones came from this. So this plant which branched, yes, it branched here, is the earliest, but when you're setting, we set from here. Ne? So early vesicular plant appears, meaning that this plant which went like this brophyte, it doesn't have a vesicular plant. It doesn't have a vesicular bundle. Vesicular bundle is xylem and phloem. Xylem and phloem. Phloem, you remember, is responsible for transportation of manufactured food. And then xylem is responsible for transportation of water and mineral salts. Now, look, all these plants, they have a vesicular bundle. All these plants, it is a seed plant. These ones, now these two, the gymnosperm and angiosperm, they have a what? A seed. They have seeds. And then these ones, flowering plants. Only this one is one which is flowering plant. So these ones, all these ones before this, all these ones, they are non-flowering plants. So these ones, we say that we call them thallus. So what is the difference between the brophyte and the pteridophytes? Let me show you here. What is the difference between the bryophyte? What's the difference between the bryophyte and pteridophyte? The bryophytes, yes, they require water for reproduction, a lot of water for reproduction. Pteridophyte, they only require a film of water, but this one require a lot of water for reproduction. And then these ones, they don't have a vesicular bundle. They don't have xylem and phloem. Yes, they are just thallus. You understand? These ones, pteridophytes, yes, they do have the leaves, but they are not that strong enough, meaning that they are still not able to stay in harsh conditions. So as they move from brophytes to pteridophytes, they are increasing in the dependence of water. So this one requires less water. This one does not require water for reproduction. We call them gymnosperm. Gymno comes from what? Gyne. Gyne means what? Yeah. If you want to be a gynecologist, what are you talking about? What do you want to deal with? Yes. So these ones, we say gymnosperms is, these ones, they have what called naked seeds. Hey, by the way, I'm not teaching. Yeah, I was just trying to tell you what I'm supposed to show you, what we're supposed to focus on. Anyway, angiosperm means flowering plants. Okay, let me not teach unless someone asks me a question concerning about, yes, let me say, animal phyla. Examinable. Yes, I'm coming there. Yes, so we are saying that you need to focus on, you must know the differences among these ones, but don't make a mistake. Can I repeat? Don't make a mistake without knowing or how to use or trying questions concerning about a phylogenetic tree, because we will bring phylogenetic tree in grade 12. So we have to inculcate, I repeat, we have to inculcate techniques how to use this phylogenetic tree from grade 11. Hence, in each and every paper, in most cases, we bring a phylogenetic tree. I'm clear. I'm saying it in your ears. Number 2, I said that on this topic, I said that I don't know if this is a medicine for people. As I always say it when I'm teaching, that don't make a mistake and you go without knowing the structure of a moss. Why? I don't know why. I'm asking us because maybe it's easy to grasp. So go back to your book, or whatever book you are using, try to read it. Don't make a mistake not to read it. Yes, why don't they bring this? Why? Is it too complicated to draw? No. But why? I don't know. But we like. Based on the analysis I've been doing, and I've been, when I sit down and then I compare different question papers and the stuff, you only find out that 99% of the papers, go to the past papers. If you think I'm lying, go to the past papers, you'll see. Why do they like this? So I'm done with that. So these are pteridophytes, vesicular plants. But these ones, the difference between the bryophytes and the pteridophytes, sometimes you call them pterophytes. Sometimes you call them pteridophytes. It's the same thing, ne? Yeah, it's the same thing. So these are vesicular. This one, they have a vesicular bundle, but pteridophytes, bryophytes, they don't have a vesicular bundle. They require a fair amount of water for them to carry out reproduction. But bryophytes, they require a lot of water for reproduction. And then when you go to the gymnosperm, we call them naked seeds. These ones, they produce seeds, but the seeds are not enclosed in a fruit. For example, the coconut you are eating is an example of a gymnosperm. I've already designated different videos concerning about this gymnosperm, bryophyte. So just go to Thundereduc or B.Saidi channel, then you'll be able to find this. We have to smash grade 11 paper. General characteristics of gymnosperms, we have talked about that. True roots, sporophyte is a dominant. Whoa, let me talk about this. I've talked about this. All the plants, they have sporophytes as a dominant generation, but the bryophytes, the first class, they have the gametophyte as a dominant generation. This one is the only one which has a gametophyte as a dominant generation. The rest, they have the sporophyte as a dominant generation. I think I'm done with this. Angiosperms, yeah. When you go to angiosperms, we don't ask you too much about angiosperms. We only ask about the structure of a flower. How does insect carry out pollination, or the different types of pollination? That's what you usually focus on. Yes. So general characteristics of angiosperms, yes, you read, but when we bring a question, we can just bring based on the maybe dependence on water. Explain this categories, the bryophytes, the pteridophytes, angiosperm, and gymnosperms. So that's what we usually ask. We can just give you one point and ask you to explain it. Sexual reproduction, know that sexual reproduction is, you don't need, you require gametes, you require two parents. Sexual reproduction, you require only one parent. You understand? So sexual reproduction, you require two parents for you to carry out this reproduction, or reproduction take place. Examples is budding, fragmentation, fission. Hey, don't confuse fission with fusion, with F-S-I-O-N. Fusion. Fusion means combining, while fission is splitting. This is splitting. So please don't confuse the two. Yeah, regeneration. So these are examples. What can I say here? Yes, you can read. Yes, you can read, because sometimes you can bring about a table for you to draw the similarity, the difference between asexual reproduction and sexual reproduction. Asexual reproduction requires one individual. Sexual reproduction requires two. Asexual reproduction uses less energy. Sexual reproduction uses a lot of, why? You have to chase someone, you have to beg someone. If you're a dog, if you're a cow, if you're a what, you see that they have to chase each other to bring about this reproduction, or this reproduction to take place. Yes. So that's why you're saying that it requires a lot of energy. Just a moment. Some technical issues here. Let me know the questions, guys. Let me know the questions, or I'll give you some time also. Maybe if you have some questions, I will smash them. Yes. Check. This is sexual reproduction. Do you know how much energy this fly, Musca domestica? Yes, we call it Musca domestica, the house fly. Is using. Requires too much energy. Sexual reproduction. Okay, I'm not teaching. I'm just talking about differences between sexual reproduction and asexual reproduction. Flower. Flower. So there are so many differences. So I'm not going to. So just go back and read about the difference between the two. Yes. Remember, this is term to what? What I've focused here more, please take as crucial. Why? Because it's the same examiner, same teachers. We have same feelings when you're teaching. Say, I think I should bring this question. This teacher is different from this teacher, is different from the other teacher, but you end up seeing them bring the same questions. If it was me setting, for example, I'm just saying, I would have brought this. And if another person, same story. Guys, please don't go to the paper without knowing the structure of a flower. These are simple things. Actually, we want I always tell people to read these simple questions because these are just simple questions. Memorize, write the answer. Memorize, write the answer. Memorize, write the answer. Yes. Videos concerning about flower, they are there on YouTube channel, Thundereduc, B.Saidi. Still, you'll find them there. So, don't go without knowing the flower. Yes. Is this insect pollinated or wind pollinated? Check this and check this. The moment these anthers are below the stigma, it means that it is insect pollinated. Because if it was wind pollinated, the anthers, they would have been up so that when they shake, the pollen grain will fall on the stigma and then fertilization will take place. But because now the anthers are down the stigma, it means that this flower is an insect pollinated flower. In other words, no, the insect pollinated flower and wind pollinated flower. I'm not teaching, I'm just trying to tell you. Hence, I'm not going to spend too much time. A combination of anther and the filament, how do you call it? The male part of the flower is called the stamen. And then the female part of the flower is the stigma, the style, and the ovary. And then the whole part, we call it pistil. Scientifically, this is called androecium, while this one is called gynoecium. Gynoecium. Yes. Anyway, let's continue. I'm not teaching, I'm just predicting. So guys, don't go to the paper without knowing this. Can you label this? Can you? Okay. What is this? 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 You must know. It must be with the you know that this petal, this is anthers, this is filament, this is what. So you have to be knowing these things. It's like a song. Don't go to the paper without knowing these things. What if you're a teacher? What are you going to teach when you don't know these simple structures? Anyway, please go when you know them. Parts of functions of the parts, meaning that you must know that the petal is this, the function. Stamen, what is it for? Anthers, what is it for? Filament, what is it for? So you have to know the structure of a flower, in other words. Pollination, how do you, here, go draw a table of insect pollinated flowers and wind pollinated flowers. Insect pollinated flowers and wind pollinated flowers. Please go when you know that. So basically, flower is done. I'm coming back here. Biodiversity in plants, put 30 hours. Remember, you have 100 hours. Term 1's work, that term 1's work, the first topic, put 10 hours. Because we have done a lot of activities, a lot of questions concerning about that. You think you expect them to bring them back that much? I don't think so. Bryophytes, or biodiversity and classification in plants, please put more. Why? Because there is grade 12 work which we want you to master before you go to grade 12. Phylogenetic tree. I've talked about it. Ne? And I've talked about the structure of a flower. And I talked about the medicine of the teachers, meaning that tablets of the teachers, they can't live without them, the structure of a moss. Let's go to the next topic now. Someone was asking me, do you expect this to come? Yes, because it's a topic, it's among. Sometimes when we don't bring the phylogenetic tree in plants, we bring the phylogenetic tree here. Ne? We bring the phylogenetic tree here. You see? Here, we can bring it there. So flower, check. What type of formation is this? Check. They're telling you insect. Insect. You know why? As I explained, check this. Anthers are below the stigma. The petals are big. Don't forget that the brightly colored petals. If you check the other flower I showed you, it's brightly colored. So we have some characteristics which we are supposed to look at to classify whether it's wind pollinated flower or insect pollinated flower. So I'm not going to go through the questions. No, I'm not going to go through. This is what I was saying. Wind pollinated flower, insect pollinated flower. So at least go when you know that. Yes. So I think I'm done with that. Ne? Yeah. So now we are going to the next question. The next question that is, when we come to the classification of animals, we like this. We love this. We love this. Ne? Yeah, sometimes I tell students what you're supposed to look at. Don't go to the paper without knowing these different layers. Ectoderm means outside. Endoderm means the inside one. Mesoderm means the middle one. Yes. So please don't go to the paper without knowing this. What about the, let me show you, there's something which I want to show you. Polyphera, look at this. So please don't go to the paper without knowing this. Ne? Yes. So I'm clear there. Then, which type of symmetry? Talk about the symmetry and talk about cephalization. Cephalization is the development of the head. Cephalization is the development of the what? Head. And then symmetry is the dissecting dividing the organism into pieces. If you look at this one, it has one hole. It's the head and is the if you want to poo, the same hole. If you want to eat, the same hole. Yes, we call them. They don't have. They don't have a symmetry. They are symmetrical. Cephalization, no. They don't have a cephalization. Development of the head. They don't have the head. Do you understand? So, if you look at nidarias, I don't know whether you call it cnidaria or nidarias. Nidarias, this one is asymmetrical. This one, Polyphera. They're like sponge. Yeah, that's why they call them sponges. Sponges, yes. And then this one, because of these tentacles, then they have what called a radiosymmetry. Meaning that when you talk about radiosymmetry, it means that you can cut it there, yes, you can cut it there, yes, you can cut it, this part is equal to this. You can cut it into different planes. Yes. So we call it asymmetrical. No. We call it radiosymmetry. But if you have something like this, for example, this side is not equal to this. This side is not, so we call it asymmetrical. Yeah. So you don't need to even memorize that much. This one, platyhelminthes. These are the ones, the flat ones. Some of you have ever gone to the toilet and you find them, you caca that long one. Yeah. So we call it platyhelminthes. People who are eating pork too much. Yeah. Welcome to the platyhelminthes phylum. Yeah. So all these ones, because they have a developed head, so they have the eye. It means that they have the bilateral symmetry, and they have they are cephalized. Bilateral symmetry, and then they have a cephalization. Dorsoventral flattened, it means that your body is flattened. Yeah, just to increase the surface area. So please, these ones, they don't ask you. If they are to ask you, we like to ask you cephalization, as slomet, and then the tribloblastic. Those are the three things we like to ask about. So please go when you know them. That diagram I've shown you, yes. I know that this is a problem to students. Yes, I know that this is a problem to students. But you see these colors, I've put different colors. These are the things we are looking for, nothing else. Yes. So these organisms are very important for pollination. They're very important for aeration of the soil, decomposition. I've taught you this, and then there is a topic. What are you supposed to focus on? Photosynthesis. Because this is a small topic, put 10 hours. So now I have 10, so meaning that now I have 60 hours. Yes. Let's go to, you see the question they're asking? It's the same thing I've been talking about. Not check. Symmetry, cephalization, number of guards, and tissue, slomet, or slum. That's what they are looking. They are not going to ask you too much things. No, they are not. So don't waste too much time. Focus on this and this. So that's why I'm saying I'm giving it 10 hours. So it's not that a big deal. Let's go to the next question. Next question is saying what? The next question, photosynthesis. Photosynthesis. Okay. Yeah. Photosynthesis. Let me show you what we like. You see now, this question. I told you that sometimes we bring the question concerning about this, the phylogenetic tree in plant diversity or in animal diversity. So, it's the same thing. You see? Pseudocoelomate, false coelom. The common ancestor. Radiosymmetry. It means this one has a radiosymmetry. This one, tissue, bilateral symmetry. All these ones up, they have bilateral symmetry, meaning that platyhelminthes, nematodes, mollusca, annelida, arthropod, echinodermata, and then codara. All these things, they have a meaning. Yes. Don't just say Cnidarias, polyphera. They have a meaning. So that's why you need to cram. You need to understand. So go and watch Thundereduc or B.Saidi channel. You'll find those videos. But for now, know how to use know how to use, sorry, know how to use the what? The phylogenetic tree. Yeah, the next question is about photosynthesis. Photosynthesis. So in photosynthesis, what you need to, just a moment. Photosynthesis. Please know what you call the dark phase, light phase. The light phase and the dark phase. At least you know where are they found, and then the structure of the chloroplast. So know the structure of the chloroplast. Where does dark phase occur? Where does light phase occur? And then now, how do you interact the dark phase, or the process of photosynthesis with respiration? Let me show you what I'm saying. Structure of a chloroplast is easy. Yeah. So I have a feeling that because we brought this in your task, I don't expect this to come back. I'm not saying that I don't read it, but based on my analysis, we can't bring it in a task and then we bring it again in exam when there is another content you're supposed to look at. So I don't think we'll bring the structure, or they'll bring the structure of a chloroplast. I don't think so. Yes. So I'm gone. Let's go to the light-dependent phase, the phase which requires the light, and the dark phase, what you call the light-independent phase, the phase that doesn't require light. What happens in light phase and what happens in the dark phase? In the light phase, yes, the first thing which occurs is the sun will be trapped by, I'm just going to summarize this, by this called thyroids, yes, the grana. And then now the function of the light, this is called light phase. Light phase occurs in the grana. You understand? While the dark phase occurs in the stroma. So the light phase, the function of the light phase, when the light strikes this water molecule, it splits it into two. How do we call that in science? Photolysis. We call it photolysis. So we call it splitting. Splitting the water molecule using light. Photo, light, lysis. Lysis, when you say lysis in science, means splitting. Yes. We call it photolysis. Then now, this hydrogen ion is going to be accepted by NADP, and then it's going to become NADPH. And then the NADPH is going to transfer this hydrogen ion into stroma. Now the oxygen is being released outside. You see? Now this one will enter. Some ATP is being formed. Now the ATP, the NAD is going to enter the NAD is going to enter. It's going to enter the dark phase here. You see? So the NAD, if it enters the dark phase, once it enters the dark phase, what happens? This hydrogen is going to combine with the carbon dioxide, meaning that carbon dioxide is being used in the dark phase, plus the ATP, and then glucose is going to be produced. So basically what happens in the dark phase is the combination of this carbon dioxide plus the hydrogen ion from the light phase which occurred in the grana, plus ATP, and then glucose is being formed. And then what happens in the light phase, we have said that what happens in the light phase is this light is going to strike the grana or the thyroids. It's going to find the water molecule there. It's going to cause this water molecule to split, a process called photolysis. To form hydrogen ion, oxygen is going to be raised as a byproduct, and then this one is going to combine nicotine amine adenine dinucleotide phosphate. That is NADP in full. Nicotine amine adenine dinucleotide phosphate. No one is going to ask you that. Just say NADIP. All right. I've never seen a question asking you what is NADIP info. Yes, here in this grade. So please go when you know how to. If you don't understand this better, there is a video which is dedicated to explain photosynthesis on Thundereduc and B.Saidi channel. Yes, so please you can visit one of the channels so that at least you are not missing out. Don't forget to use this distinction material. There is no question you should gonna find and you can't answer with this distinction material. So this is basically what happens. Split, light is absorbed, water is being split under the influence of light, and then oxygen is being produced, and then the NADP H is going to go to the dark phase, and plus ATP is going to go to the dark phase, and then now what happens? The ATP is going to be split into ADP, so that you can go back and pick another energy, and then also this NADP plus is going to release the hydrogen here, and then it goes back to this. So that's the cycle. But now you have to know, how do you interconnect this with the respiration? So the difference in light phase and dark phase, please go when you know that. At least you, yes. There is this experiment we like most. I don't know why. Yeah, we can ask you that question, but not that much. But we like that one of light. Where is it? We like that question of light. Yes, this one. So, yes, we have different versions in which we can. Let me show you. Different different versions in which we can set this. Let me show you. Let me show you. Let me show you. Let me show you in this booklet. Yes. Yeah. Yeah. We like also. We can set it in this version. Yes. Or we can set it in this version. So it's the same thing. So it depends on, this shows how light is being used to produce, the what? The oxygen. And how do you calculate oxygen? You calculate oxygen by this and this. And don't forget, we also brought this, I think, yeah, in the toast. Yes, but we like it also. So please take note of that. Then, what terminology are expected? Akira is asking, what terminologies are you expecting? Terminology. You see, when I keep on talking about, these structures, I talk about those terminologies. I keep on talking about terminologies. Like I talked about a bacteriophage. What is a bacteriophage? Is a terminology. I talked about immunity, vaccination. I talked about a vector. We like also to ask a vector. What is a vector? A vector is an organism which transfers the disease-causing organism. It does not cause a pathogen is a disease-causing organism. You understand? But a vector, like a mosquito, let me just explain using malaria. A mosquito is not the one which causes malaria. No. Mosquito carries a disease-causing organism which causes malaria. For example, Plasmodium. So a mosquito is a vector. It carries a disease-causing organism, which is the Plasmodium. Then the Plasmodium is the one which is called a pathogen. So you must think. We like to ask pathogen, vector. Why? It's because students confuse them. Immunity, vaccination. We like to ask that because students confuse vaccination, immunity. You see, we like to ask those things. Yes. So as I'm talking, like now, I've talked about this. I was talking about these processes. Now, which terminologies do we usually ask here? factors affecting. Here we can talk about the dark phase. Yes, will bring about the phase of photosynthesis which occur in absence of light. It's a dark phase. The phase of photosynthesis which occur in presence of light is called what? Light phase. So you have to know those. You have to know the science behind. We can also ask you maybe a question. The process by which, because I said photolysis, the process by which light breaks down water molecules in photosynthesis. How do you call that process? Photolysis. Yes. So we can ask you different questions, but make sure that there are these terminologies which are always there on the tips of the greenhouse effect. Yes, we can also bring that as a terminology. There are these terminologies which are always on the tips of the what? Of the examiner. I must bring this. And what that person is thinking, I'm also thinking about it. That's why you bring it. Yes. So, okay, the next, this topic, photosynthesis, you can give it 20 marks. 20 marks. I'm saying 20 marks, 20 hours. I'm splitting, okay, 10%, 30%, 10%, 20%. Because when I say 20 hours, someone can say 20 hours. No, I'm saying I just give an example of you have 100 hours. You have 10 hours for the first topic, 30 hours for the second topic, 10 hours for the third topic, 20 hours. You see? Yes. I think Akira, clear there. Yes. Then when you go to the next question, is gonna be on animal nutrition. Obviously, when you talk about, there is no way if we bring here, we like three things. Obviously, the structure, this structure, you must know it. Yes, digestive system, you must know it. We like the villus, the structure of the villus. Yeah. People have been teaching you know what I'm always saying. Villus. Don't go to the paper without knowing it. Villus, the structure of the villus. Make sure that you know it. Yes. So, how is these questions being set? This one, peristalsis, different types of digestion, mechanical digestion, and then chemical digestion. Yes. Cool. No, when is mechanical use of energy is mechanical. Chemical digestion, use of enzymes, meaning that in chemical digestion there is involvement of enzymes. In mechanical digestion there is no involvement of enzymes. You use energy, hence you're supposed to know that. The different enzymes, you can also master them, but we like to ask you this. Yeah, and you can check in most of the questions I've been giving you. You'll find that villus is always there. I can't skip it. And also this question, but the problem is, this the negative feedback mechanism. Do you know why we bring it? It's because it's also grade 12 work. I'm not saying that it's coming. If it doesn't come now, it will come tomorrow. So make sure that you go to the paper when you know this, the negative feedback mechanism of this glucose. Negative feedback mechanism of glucose. And then the structure of a villus plus this peristalsis. Peristalsis is also another terminology we can ask. Yes, the rhythmical contraction of the muscles. It's a reflex action is triggered by presence of food in the alimentary canal. It is important to transport food. Yes, this is also, we like to ask you this. Breakdown of fats or lipids into small particles. Emulsification is also another terminology we can ask you. So I think I'm clear here. Villus, I repeat, don't make a mistake going without knowing it. what terminologies are. Okay, I think Akira, I'm clear there. Please don't go without knowing the structure of a villus. Goblet cells, yes. Goblet cells, these are cells which secrete mucus. Yes. So basically, they bring this so that is protection. Protection. Yes, I said this. Please, this. Please go when you know it. The negative feedback mechanism. Keeping internal environment constant, we call it homeostasis. Yes. And then when you talk about the negative feedback mechanism is bringing a situation back to normal. For example, you are hungry. What do? You eat. It's a negative feedback mechanism to remove the hunger. How about when you are full? You go to the toilet. When you come back from the toilet, how do you feel? Very good. The best moment you can have ever experienced. Hey, is a negative feedback mechanism to bring back the situation to normal. If you are pressed, how do you stand? You are going to the toilet, you find someone in the toilet. You are depressed. How do you feel? After peeing, how do you feel? It's a negative feedback mechanism. Yeah, you feel good. I know, you feel good. Yes. Best moments ever. Anyway, please go when you know the negative feedback mechanism of this glucose. I'm not saying that. It's the one. Diabetes can also be another terminology. So, can be. Is there any other question? I don't see any question here. If you have a question, let me know. So, if you have a question, let me know. So, we are saying that this one is not going to come. Don't bother. Yeah. Okay. It's because it's cooked out of the syllabus. Let's go to cellular respiration. Cellular respiration. I'm not teaching. I'm just highlighting. So I'm not going to be teaching too much. Unless you have a question that I don't understand this subtopic, then you let me know. Yes, in the comments. But if you don't have a question, yes, I'm just going to highlight what you expect to find. Cellular respiration. So I can say that digestion, you can also put 20%. Yeah, these are, why am I saying the 20%? Because these are term to work. Don't expect to bring too much of the virus and the bacteria and what when you have already set them. We know you know them. We have to bring these ones you have just studied and then see if you know them. So this is the only chance to set them. Hence, photosynthesis, digestion, and respiration. Make sure that you go there when you know them very well. As the same way I've tried to highlight for you, just to show you what do you expect to find there. What are the things you expect? Let's go to respiration without wasting time. Cellular respiration. Anyway, have you ever brought this diagram? No, because we saw the diagram of this is a mitochondria, the powerhouse of the what? The powerhouse of the powerhouse of the cell, meaning that that's where power is being generated from. It's the engine of the car. That's where the what? So these are all organelles, chloroplast, and then this one mitochondria. But chloroplast, I have my bias on it for now. Why? Because they brought it in the task, so I don't think they'll bring it. So put more effort on the chloroplast. I'm not saying that is coming, but I'm saying put more effort on the chloroplast. So when you go back now, no, not mitochondria, and then when you go back to photosynthesis, you know where I said they like to ask this dark phase, light phase, um, this experiment, the experiments I showed you where to focus on. So I'm not going to go there far. Let's go to this. So outer membrane, inner membrane, I explain it in a video so you can find it. Process of Krebs cycle. I think, should I explain this? But I explained it. Okay, just in 1 minute. Let me just try to show you Krebs cycle, oxidative phosphorylation. Okay, let me show you. Okay, let me just see here. Guys, don't forget that you will have one experimental question, né? Independent variable, dependent variable. If I've forgotten, please remind me. So this is what we are talking about. Yes, you see. So limiting factor. We can also bring that as a terminology. Limiting factor. A limiting factor. It cannot be in excess. Never. Whenever they say is in excess, means that is a limit. You see now the other question I talked about. Yes, we like it. We like it because we can't live without it. This one I said I have a bias on it because we brought it in a task. So yes, let's go to digestion. I talked about it. I said don't go without knowing this because these are basics. At least you must have 2 diagrams in each topic you're supposed to focus on. Yes, so you can't go without knowing the naming of these things. Eh? Yeah, where does this reaction takes place? Bile. Yes. So, structure of the virus. Please don't go to the paper. Yes, I've forgotten about something. The one we like to ask about the virus. How is this virus adapted to its function? Here are the answers. Here are the answers. Here. This one. Small intestines. Yes. Not that one. Here. How is this virus adapted to its function? Epithelium is only 1 cell thick. Goblet cells. Mitochondria supplying energy. For what? When it asks you adaptation, you give, for example, epithelium is only 1 cell thick. For what? Allowing nutrients to pass through quickly. So 1 cell thick nutrients, they pass through quickly. You see, goblet cell mucus to moisten the surface and also increase the dissolving the substances. You see, mitochondria energy for what? Active absorption. Microvilli for what? Increase the surface area. Lymph for what? Transport lipids. Yes, please. Whenever we ask you, because you can't just bring a diagram and then just let's go to this question and this. You can't. Let me see if they didn't set a question here concerning about adaptation. Yes. They're saying that what structure would you expect to find the microvillus? Yes. So yeah, even this one is also related to the adaptation. So is absorption of glucose or amino acid active, transport or active or passive? It is active. Why? Because it requires energy. So you see that mitochondria is needed. So let's go to respiration, and I'm gonna go through check again. They have brought it back. The structure of what? Structure of alimentary canal. So again, born. My question I was looking for. Bah, yeah, bah. Is a slap in the face. Explain 3 structure adaptations mentioned in the go on. 6 marks. Mahala. 6 marks. 3 points. 6 marks. Mahala. We give you a tick there. Give you a tick here. Give you a tick there. We give you a tick there. You see, 3 points. 6 marks. Mahala. Don't go to the paper without knowing. You see that we don't have new questions. I'm telling you, we don't have a what? New questions. These are the questions we bring on. Questions we're going to set, we have been setting, we will set. These are the questions. Let me be specific. We have been setting. We are setting. We have set. We will set. Those are the questions, meaning that they are not new. If you are just reading without practicing questions, you are in a wrong class. You have to maximize questions they have given you. Let's go to the next question, that is respiration. So respiration, what you're supposed to focus on, I think I've spent too much time there. In the remaining few minutes, respiration, what you're supposed to focus on, look at the structure of the mitochondria. We have seen it. And then talk about these phases. Glycolysis, Krebs cycle, and then oxidative phosphorylation. Then after that, talk about the differences between this and this. And then lastly, we also love to bring this, but if we are to bring this, we bring it in an experiment. Yeah. So now let me just explain this. Let's explain this, because sometimes, okay, Krebs cycle, or this process of respiration, so also expect you to have 20% in our respiration. So let me show you. Respiration is divided into two. It's divided into what's called aerobic respiration and anaerobic respiration. Aerobic respiration requires oxygen. Anaerobic respiration requires no oxygen. So when you look at this, where do we use oxygen? We use oxygen in these two phases. You understand? So because we use oxygen in these two phases, yeah, it means that this phase does not require oxygen. It means that anaerobic respiration only occurs in the glycolysis. It takes place. Anaerobic respiration only involves glycolysis. It doesn't involve Krebs cycle and oxidative phosphorylation or transportation of electron through a chain. So what happens? Glucose is a hexose. Hexose means 6. Hex. I explained this a long time. Hexose. So this one means sugar. Glucose. Lactose. So ose means what? A sugar. So hex means 6. It means that 6 carbon atoms. 1, 2, 3, 4, 5, 6. So 6. So during glycolysis, it splits it into 2. 1, 2, 3. 1, 2, 3. And these two molecules, we call them pyruvate or pyruvic acid. Hence here, we're saying that is divided into two molecules, 6. And then during this process, hydrogen is being produced. And this hydrogen is going to be captured by NAD. So it's going to become NADH. Don't forget that for respiration is called NADH, while for photosynthesis is called NADP. NADPH. Yes. So please don't confuse the two. So now the NAD is energized the molecule which enters the chain. Yes. Where does this one occur? It occurs in the cytoplasm. Yes. And then now these two, they enter the mitochondrion. So when they enter mitochondrion, yeah, it undergoes chain of reaction, and then monad is being produced, and the FAD, H2. So these two, they also join the chain, and then they join the electron transport system. So as they move, like it's like a ball, you have different people, different people, different people, different people. These are boys, ne? And then this boy has a ball, gives it to this one gives it to this one gives it to this one gives it. So as this one gives a ball, something is lost. As this one gives this one, something is lost. As this one gives something is lost. What is lost here is the energy. You understand? Which called the ATP. You understand? So ATP comes from ADP plus an inorganic phosphate. So ADP is adenosine triphosphate. Adenosine triphosphate. So adenosine triphosphate. And then this is called what? This is called is there any question? Can you please teach us a bit about biodiversity of animals? Maybe you are not in class, but I've tried to highlight. You can watch the video just a bit. Just what you're supposed to focus when you are studying, what you're supposed to focus on. I tried to explain. Yeah, Maseko. I think I tried to explain it. Yes. So, and also, if you don't understand, there is a designated video I try to create. Just go to Thundereduc. There's a designated video I try to create, so you can get it, so that maybe you are able to understand the concept. So I was saying that. So this is NAD, adenosine diphosphate, and then a phosphate. So it di means two, and then phosphate is just two phosphate. Adenosine, adenine, just adenine. I think you know adenine. Di and two phosphates plus this phosphate to form triphosphate. Hence, you form ATP. This is the one which is being what explained here. Yes, that ATP is being produced. So it means that what happens, this reaction, ADP react with inorganic phosphate to form ATP. So in each process, ATP is formed. ATP is formed. ATP is formed. At the end of the day, you'll find that you have almost 32 ATPs, 36 ATPs or 32 ATPs, depending on if you put in the capital or you subtract the capital. When I say subtract the capital, the one which you used at the beginning, because you had to be with, it's like a matchbox. You can't have light without matchbox. You can't have fire without a match. You have to first light the match, and then you get more heat. Yes. So I think, yes, you need to focus on this. There is also a designated video concerning about this respiration. So make sure that you get to know this. Wrap up. Those people who are not here, let me wrap up, and then I conclude. Please don't go without knowing how do I identify independent variable, dependent variable. How do you identify independent variable, dependent variable? And then how, if it's a table broad, how do I identify that this is independent? The first column in most cases is independent, the next column is dependent. Use ID method, independent, dependent, XY. Independent on the x-axis, dependent on y-axis, meaning that this is going to go on the y-axis and then this is going to go on the x-axis, just in case they brought the graph. And then you will never have a graph like that. You will never have a graph like that. If you find this graph, just so that you swap to the axis, and then this graph is there, that graph is there, so this and this you can't find. We mark for the scale, make sure that if just the graph is there, 2, 4, 6, the range is the same. Don't say 1, 4, 5, the range won't be the same. It must be the same. And then also make sure that what you put here, you put units, and what you put here, you put units. The units are always given. Yes. And then you must always put a title. If it is a bar graph, I have a full video concerning about this also. If it's a bar graph, make sure that these bars are the same, and even the spaces are the same. These spaces are the same. The bars, the size of the bars are the same. Yeah. If you have two, like you have a table, it has two columns. You have X, Y, and Z. These two, they are going to be plotted versus this, meaning that you first plot this and this, it forms one graph, and then this and this, you form another graph. You understand? This and this, one, this and this, you form another one. So make sure that at least you know. How do you increase the reliability? Reliability is increased reliable by repeating the experiment several times. Or in the experiment, they can say that they repeated the experiment or they did it 20 times. That is reliability. How did they increase the reliability? They repeated the experiment 20 times. Or increase the sample size. Sometimes you can say that they used 200 people. So that is reliability. They have used a high number of people. Please don't go without knowing that, because that question will always it will always be there. Yes. Then lastly, as I conclude, I said that now I'm just going, maybe I'm going to use 2 minutes to conclude. I said this. Okay, let me start from here. This, I explained it. I said that this is term one's work. This topic is term one's work. But we like to bring a virus. Make sure that you know the characteristics of the two. So those people who didn't note down what I was saying, make sure that you have they know the characteristics of the virus, the living characteristic and non-living characteristic. Yes, I say this can be as a biological term. A virus attack bacteria. Yes. Then bacteria, structure of a bacteria. This can be brought where the different types of bacteria shapes. These ones, we can bring them in the terminologies. Protista, yes. Protista. Eukaryotic, we can bring eukaryotic. U means true nucleus. They have a true nucleus, yes. And then this one is protista. Phytoplankton, zooplankton. Phyto comes from plants. Zoo is a zoo animals. Zooplanktons can also be autotrophs, heterotrophs. We can also bring them as a terminology. The person who was asking for the terminologies. Fungi, we say that, yes. Another diagram, the third diagram is this fungi, bread mold. Guys, this diagram I'm telling you, those are the diagrams we bring. This one, I'm sure that 70%, because we don't bring all of the diagrams, but the 70% of the diagrams I've told you, you will find 70%. Why? Because we don't have any other diagram. Yes. So please know the bread mold. This other one, not too much. This one, it was too much time. Yeah. Bread mold, we like it. Go look at the different questions. You'll know. You'll tell me that what I'm telling you. If we don't bring this economic importance of fungi, I said we bring the immunity. Yes. But don't forget that this is term one's work. That's what I said. Yes. Then we got to mutualism. These ones can also bring commensalism. We can bring them in terminologies. Yes. Diseases. This one must not waste too much time. Something happened. Just a moment. Technology. Technology. Just guys, a moment. I don't know what happened, but I think with distinction material, you Okay. Okay. Yeah. It's here. Yeah. It's back. I don't know what happened. Oh, it has come back. I think it was error with memory. Yes. So I said that these ones we are not looking at. Don't waste too much time with these diseases. Yeah. You know why? I said that we teach different diseases. I showed you even the different questions I was explaining. So just know how it works. This one, I say that immunity, focus on it because we like it, because we know that students don't know, with the student being confused with this. Ne? Yes. Passive immunity, active immunity. So please go when you know that. So the diseases, so I think I'm cool there. Biodiversity of plants, I say that this one, because it confuses students, make sure that you know this. Why? Because this is great for work also. That's why we have to bring it. If we don't bring a phylogenetic here in these plants, we bring it in the animals. So yeah, so I've explained that. I say that this is a medicine for teachers. Tablets, like they this is they can't live without it. Yes. So please don't make a mistake with going without knowing it. Then we say that this one, yeah, we said the properties. We say the characteristics. These ones are naked seals. These ones are true. They have a fruit, meaning that the seeds are enclosed in a fruit. Then I said that in the angiosperms, asexual and asexual differences, you must know them. I showed you the questions. And then also the flower, talked about the structure of a flower and the difference structure of a flower. I explained and I ask you how do you identify this is insect pollinated, this is non-insect pollinated. You see, I explained it. And then I also talked about the insect, the difference between insect pollinated and wind pollinated. Please make sure that you know that. Then when you go to biodiversity of animals, we say that phylogenetic tree, still take note of that. And then also we like this. Please don't go without knowing it. And also know cephalization, symmetry. Yes, as either is asymmetrical, bilateral symmetry, radial symmetry. I explained it. Please don't go without knowing it because those are the questions we ask. We don't have time to set new questions. I'm just saying it in your ears. Pretend that you didn't hear. We don't have time to set new questions. What do? We use past papers. So what I'm saying is use past paper questions. You'll be able to pass. Yes. I showed you the different questions. Platyhelminthes is fertilized. We talked about all this. So here, what you need to do is know which one is fertilized, asymmetrical, xylem, and then number of guts. That's all. We can't expect more than that. Photosynthesis. I said I'm biased with this. You know why? It's because we brought it in, so I don't expect it. If it comes, it will come 5%. So I'm not saying that don't read it. That I said don't read it. I said 5%. That is my feeling. I'm just sharing what I'm having in my heart and based on the analysis I've done. So I'll also do. When are you writing? You're writing. Yes, I should. I think I should do it tomorrow because you're writing. I think it's 2 weeks from now. Yeah, when I do it tomorrow. Some people they say that that I have the paper. Who told you? I don't have. I'm not the one. But I use the principle of analysis. Yes. Okay. Here. I say that make sure that you know how to differentiate between light dependent phase and light independent phase. I said that even here there is another terminology. I said it here. I said it here. Is here. Where is it? Where are you? Okay, I think the person who was asking about terminologies, I talked about it when I reached here also. Yeah, it's here. We also bring another terminology. Radiant energy causes the water molecule to split. Photolysis. You can also bring another terminology there. Yes, ATP can also be another terminology. Yes, so make sure that you know the difference between light phase and independent dark phase. I said please. These graphs, limiting factor is also another terminology we can bring. Limiting factor. So make sure that also you know it. How do these factors affect the rate of photosynthesis? Why? There is another factor. There is another terminology we can talk about here. Denaturation. Yes, because here there is the enzymes have denatured. These ones, yes, we bring them, but not too much. We bring this. We like this, but we have different versions. And then we bring this also. Make sure that you know it. I showed you. This one is also another terminology. Yes. And then this one, virus and the adaptation of a virus, we also bring it. We like it. And also, we like this because it's a great way of work. Yeah, the negative feedback mechanism. And yes, and then, yeah, then different types of structure of a mitochondria. I said I have no choice here because we have never set it before. So that one, make sure that you read it. Here, the process of glycolysis, Krebs cycle, and the oxidative phosphorylation. Please don't go without knowing it. I try to explain it. If you don't understand better, there is a video concerning about that. Just look for Thundereduc. And then this one, we can bring it also in the practical way, but I'm not so sure. Yeah, because you have a lot of content for fewer marks. 150 is fewer marks for all this content. Yes. So I think that's it. Thank you very much for joining me. Don't forget to subscribe if you want the distinction. Always use this distinction material. After the paper, you'll come back and tell me about the distinction material. Come back and comment about the distinction material. Yes. And tell me, how did you find it? Yes. Did you find the paper good? Those people who are using the distinction material, did you find the paper good? Or the paper was not that good? Ne? Yeah, you must tell me. Ne? What did you find? What did you find? I love you guys. Ne? I love you guys. See you again. Ne? Don't forget to subscribe, like, and share to others for our daily new videos. Hey, by the way, people don't know that we do all the subjects. We do all the subjects. See you again. B.Saidi.